Of you. It is a great pleasure to have you here, and we are hoping to have a very productive and pleasant week together. So all of you, or most of you, have already seen this poster, probably. I would like to give you a brief overview of what we are trying to accomplish here and go into details of what has been presented already. So this workshop, as you know, is being jointly organized between two groups. One is the Theoretical and Computational Biophysics group, shortly known as TCBG, with the leadership of Professor Klaus Schulten. And the other one is the National Center for Multiscale Modeling of Biological Systems here at the University of Pittsburgh, which I'm directing. So both of those two initiatives, TCBG and MMBIOS shortly, are biomedical technology and research centers, or our activities are being funded by the program, NIH program, for funding biomedical technology and research centers. A major goal in our centers is to build methodology to develop new software. But equally important is to disseminate, to share what we are doing. And this workshop is exactly organized within this frame. So let me give you a few more details on TCBG. Has been founded in 1989. At the University of Illinois at Urbana-Champaign. And it has been founded by Klaus Schulten. Their motto has been bringing physics to life. And I can't think of a better description of actually what we are trying to do. We're trying to understand life processes with the help of physical sciences. Physical science means all the basic principles of physics, chemistry. I don't think Klaus really needs much of an introduction. He's already very well known, and we are pleased and honored to have him here. I just wanted to say, to bring to your attention a recent publication of Klaus, which turns out to be in collaboration with one of the participants here, on the resolution of the chemical structure of HIV capsid. And this is one of the most important recent accomplishments, which became the cover of Nature in the last issue. This is one important aspect of what Klaus is doing, making very significant contributions to advancing science. And the other is exactly the type of workshop that we are offering right now. And all of you, I'm sure you're familiar with NAMD and VMD already, and you will learn more. Perhaps the best measure of the impact this software has made already to the field is to see the number of citations, thousands of citations by many different users. And not surprisingly, Klaus was the recipient of the Distinguished Service Award of the Biophysical Society in 2013, and we warmly congratulate him on that. Of course, the other very important contributor to the development of the software and also dissemination of knowledge has been Emad Tashkhurshid, originally being a key member of Klaus' lab, who has now his independent lab. You can see Emad here in the middle with his lab members. He's currently an associate professor of biochemistry, biophysics, and pharmacology at the Department of Biochemistry and the Department of Pharmacology. And he is also a scientist at the Beckman Institute for Advanced Science and Technology. So Emad has been a major research area of Emad has been actually membrane proteins, understanding the dynamics of membrane proteins, the effect of lipid bilayer or water molecules. And this is just an example from a recent study of his, which just appeared in Proceedings of the National Academy of Science, where he's emphasizing the significance, the importance of hydration in enabling the transport phenomena of transporters. And we are really very happy to see him providing first-hand information here on day 3 on membrane proteins, how to simulate them. No, there's no need to say membrane proteins are the most important drug targets being located on the membrane. And it's critically important to understand how they function. These are moving targets, if you like. We need to understand their dynamics. So that is about the TCBG. Let me also briefly talk about MMBIOS, the center that we started here between the University of Pittsburgh, Pittsburgh Supercomputing Center, Salk Institute, and Carnegie Mellon. So it's a joint initiative between 3 institutions in Pittsburgh plus Salk Institute. The major overarching biological theme in our center is to understand the spatial organization and temporal evolution of signaling systems and events, with emphasis on neurosignaling. So that is what we are trying to do with the participation, with the leadership of the University of Pittsburgh and participation of Salk Institute, Carnegie Mellon, and finally Pittsburgh Supercomputing Center. So what we are trying to do, as the name indicates, multi-scale modeling, we are trying to bridge across scales from small molecules, and this is an example of transporter molecules from a glutamate transporter seen from the extracellular region surrounded by water molecules, to cellular architecture. And here you can see dendrites and spines and how they interact with at the near synapse with the astrocytes. And finally, we would like to move on to having some kind of molecular or cellular explanation for larger scale neural circuitry. And as you can see, this is quite an ambitious project. And trying to understand the molecular basis of many events has been a major, understanding the mechanism is a major goal, not only in our center, but also at the TCBG and many other labs. So in this respect, especially in our activities focusing on molecular events and how to map them to cellular, subcellular microphysiological events, I think the goals of the centers are very well complementing each other. So here is a view of the time scales of those different processes. Here you can see that we're talking about nanometers. Can you see? And here we have, each dimension is of the order of micrometers. And here we move to tens or hundreds of micrometers. So these are totally different scales. And the processes that happen at those scales are different. Within the scope of this workshop, we will essentially focus on the molecular events and perhaps how large systems, also molecular machines, behave. Our training and dissemination efforts are being coordinated by Marcus, whom you have already met with. And he will give you a short overview of some important logistics shortly. And our major goal is, again, one of our major goals is to train and disseminate future scientists. We have many software that are made accessible, and I'm not going to go into the details, but perhaps the most important one where we are trying to integrate all existing software is ProD, which will be the topic of 4th day workshop today, this week. Dr. Bakan, who is a PhD in computational biology from the joint program between Carnegie Mellon University and University of Pittsburgh, is the main developer of Prodi, and you will have the chance to hear from him directly the details of his software. It's amazing how this Prodi, as the name indicates, it's mostly focusing on protein dynamics, but now we have also extended it to sequence analysis and doing other type of functions. This is just statistics of its rapidly increasing usage and the cumulative downloads. It's being worldwide used, and there are more statistics on its documentation, etc. So essentially, this is a software written in Python that is being very broadly used, and I hope you will enjoy learning about it. Another instructor in this course will be Dr. Tim Lezon. He's a physics major from Penn State University, the laboratory of Giant Bonnevere, and he's currently an assistant professor at the Department of Computational and Systems Biology in our university, and you will enjoy, I'm sure, meeting with him too. So here is an overview of the workshop program. I'm not going to go into the details because it is, I think already Klaus will present, give an overview as well. And we are going to present overviews in every single session. Today we start with an introduction to protein structure and dynamics by Professor Schulten. And tomorrow it will be the statistical mechanics of proteins, force field parameterization by Klaus and Emad. The third day, it's going to be the simulations of membrane proteins by Emad. The fourth day will be on the introduction of collective dynamics and how we deal with it using elastic network models by myself, Tim, and Ahmed. And finally, the last day, we will show how these things can be integrated to do, in particular, druggability simulations, and also combined with information from sequence evolution. So we have several tutorials that are accessible, and I wrote here the two major websites where you can have access to the tutorials. And I'm giving examples of tutorials that are accessible there, probably the most important ones, NAMD and VMD, in days 1, 3, and Prodi and NMWIS. As you probably know, VMD is a visualization software, and so is NMWIS. And this, last but not least, I would like to acknowledge the contributions of several people. We have first of all, 4 teaching assistants will be helping us, and you may have already met Danielle here. Juan and Danielle are coming from the group at the University of Illinois, Urbana-Champaign, and Anindita is contributing from our department. You may have already exchanged emails with David and Marcus for the registration or other processes, and also some administrative assistance has been provided by Jesse and Pat. Here are their email addresses in case you need to contact them. And with that, I will just want to say, of course, I'm also acknowledging NIH. Without support of NIH, we wouldn't be able to accomplish many of the activities we are engaged in, and certainly not this type of workshop. So thank you very much for being here. And we are hoping that together we will be experiencing a very pleasant week. You will learn a lot, and I'm sure we will learn at the same time a lot. So with that, I would like to invite Marcus to give you a few very practical instructions, important ones.
So hello everybody, welcome to the hands-on workshop on behalf of the Pittsburgh Supercomputing Center. So I'm going to turn it over to Dr. Schulten in a second. I just wanted to tell you about a few things that might be useful for you to know with regards to PSC. So we'll always be in here in this room in the mornings for the 5 days of the week. The hands-on sessions in the afternoon, actually, a computer lab just right behind the door. So we'll be in there in the afternoons. You will have a light lunch served that's outside this door on the left. There's a little room on the left. The one request I have, please, no food in the computer lab if at all possible. We'll always try to keep it as clean as possible. So if that would be appreciated. For those of you who need access to the wireless, it's a little bit more complicated than it perhaps should be, but in any case, there's somebody here who can help you get hooked up to the wireless. So we can either do that for a few of you quickly now, and Dave will also be here during the lunch break, so you'll definitely get connected sometime for the next couple hours. Then the last item I had before I turn over to Klaus is, so some of you had indicated that there was some interest in presentations, giving presentations, research presentations. And so the plan was to do these tomorrow night, I think starting at 7. And so for those of you who still are interested in doing that, maybe just send me an email with maybe your name and the title of your presentation so I can start assembling a list of talks that we'll have tomorrow. And I think keeping it to 10 minutes is probably a good idea. I think we had at least 10 interested people, so 10 times 10 minutes is already going to be a long evening. So I think 10 minutes is probably a good time. So if you want to write me email, my email was on the slide, so it's just Dittrich, DITTRICH @psc.edu. Maybe just send me your name and title. Maybe you can put in the subject hands-on workshop or something like that so I can find your email easily. And I'll be here most of the week, some of the time, so if you have any questions, feel free to talk to me, or Pat, who you've all met, can hopefully help you with any questions that you have. So, okay, that's all I had to say, and I'll turn it over to Dr. Schulten. Thank you very much for coming, Klaus. Can I clip that? So it's for the camera, actually. It should be fine. There's a pointer here. Great. Yeah. Can I just clip that to your... Need some water or something?
I make a little interruption. Okay, so welcome everybody. Yvette made it very easy for me. I can actually shave off a few minutes of my own introduction, and that will give us a little time to introduce each other. But before I do that, I just would like to state that I have been and my co-workers have been working with people in Pittsburgh for a very long time. I think this is probably the 5th workshop we do at the Pittsburgh Supercomputing Center. Yvette and my group have been collaborating and writing papers together. Marcus was actually a graduate student in Urbana before he came to Pittsburgh. And the Pittsburgh Supercomputer Center is clearly one of the best centers for computational biology. We used it a lot. So it is for me extremely natural to be here, because it's a place where computational biology is really pioneered and supported and developed in the country. Yvette told already that she is directing an NIH Technology Research Center, and we are doing the same in Urbana. And so we are colleagues as directors of these centers that develop, in our case, technology for software technology, and for biomedical computing. And the software is only as good as it is being used by people. And so not only develop the software, but to also train people in the use of the software is really very important to the Pittsburgh Center as well as to our center. And so this workshop is really a very important part of us trying to bring the latest technology to the field of biomedicine and biology. And so we are really glad that we got again many people interested in coming to this workshop. We are sort of little proud in this, and when I say we, I always mean our colleagues here in Pittsburgh, and also we in Urbana, that we are really making a very strong case that it's not only the physicists who bring the latest technology to their field, but it is today also biologists who are doing that. So some of the software that we are using nowadays is really extremely advanced, and so we are doing as well, or I would say in many areas better than those famous physicists from CERN in doing our job. And just an example, Yvette mentioned it already, but she didn't present it quite right because she's too nice and too modest of a person. The latest technological advance led actually to the solution of the structure of the HIV capsid. And where Yvette went wrong is not to really point out that the main leaders of this project are actually here located at the University of Pittsburgh Medical Center. And we were lucky that they took us and integrated us, because we could bring the world's greatest computer technology that was needed for the project. But anyway, so this is also another example of the collaboration between Pittsburgh and Urbana, and also really a great example for the use of the latest technology, where I would say the biologists are clearly ahead of the physicists. We use the same computer, but we are there all the time. Our programs are running. We got already the first Nature paper out, just doing the test phase of the computer. And so I think biomedical research is where it is today. It used to be physics in the last century, and it is biotechnology and biology in this century. Okay, good. So Yvette introduced already the program, so I just would like to add just a few comments. So these kind of workshops are very dear to our heart, and we have them at least twice a year. And I always show a picture of the last workshop. The last workshop in this case took place actually in Urbana. So this is actually our kitchenette in our area, and we have a nice area where we did the workshop. And so you see there the last group picture that we took. And so this workshop, of course, we also have another group picture, so that we're all reminded of the fun we had together. And we put together a few pictures then of beautiful Pittsburgh. And to just say that we might want to enjoy a little bit also the surroundings here. Yvette introduced us already, so I want to just continue then. The program is constructed as follows, that we have in the morning lectures. The lectures introduce the concepts behind the software, the algorithms used in the software, and give examples of the use of the software. These are huge programs on the side of Pittsburgh and on our side. So we can, of course, do this only in a very exemplary fashion, but that is basically the idea, that we are really telling you something about the programs you actually use, because we don't want to train monkeys, that I just tell you where to push the button, but rather I want to tell you what the programs are actually doing. And I can tell you already now, most of the questions you are going to ask me will be answered with a simple verb, think. You know, like not, oh, you don't ask me recipes, cooking recipes or something. I try to tell you how to think about what these problems are doing and how you should approach the problem, rather than that you are getting away here with foolproof recipes to cook, to make a nice pizza or something. In the afternoon, we have then the hands-on session. The hands-on session are by far the most important part of the workshop. And so there you work yourself through the tutorials. The tutorials are all available on the website. So in principle you can do everything at home, because the tutorials are actually written for self-study rather than that somebody sits next to you. But there are two reasons why you do them better here in the workshop. And the first reason is self-discipline. At home you always go and have another cup of coffee, or a friend calls and you go to the movies. Here you work even to the end of the tutorial, and it's at the end is often the stuff that is useful, a little bit harder to learn, and so you might reach the end here and then become a better person in terms of computational biology, and without coming here you might never reach that high level. And the other reason is, of course, that you can ask people. So we have the lecturers will be here available for you, but also the teaching assistants, and so you can ask then lots of questions, and you can actually also ask each other. It would be great if you help each other with some of the maybe more mundane things, so to make things roll as quickly as possible. But the questions are very important. I might just add here that you can, of course, and we encourage that very, very strongly, also move already to your own projects, and there we can help you too. And so I myself, I always sit like for many hours with almost every workshop participant who comes to me and wants to get some advice on their particular project. And so I'm very glad, and I know that Yvette and Emad will do that too. I actually have to say that Emad would be here. He's really even more eager than I teaching this, but they only arrived last night at 4 o'clock. And I was lucky, I got a later flight and arrived early, and they got an early flight and arrived extremely late. So they will be around here, but I think they deserve a little bit more sleep. So the other faculty will also be available for helping you with your project. So the best is that you tell me, oh, I would like to talk to you, and then I make a little list of names, or I try to remember, and then I schedule half an hour or so with you to discuss your project. And I'm really glad to do that. So this is sort of like the way the program is structured, and I can then go already to the end, and like to make a few comments here. That the course is a volunteer effort, so really thought of course, it's a good thing to do to develop software and also train people in the software. But if we wouldn't train, probably we would still get our funding for the technology development. And so it is really like we believe in the training, we want to do it, but we do it really, how should I say, like as volunteers in a way. And so like I like to point that out. So the hands-on sessions are very important, I said. Then the aim is that you do, the emphasis here is on the word do, that you do computational biology. So I'm a theoretical physicist and asked myself for many years, how can you teach computational biology as opposed to theoretical biology? And there is a difference, and the difference is that computational biology is sort of halfway between theoretical biology and experimental biology. In a way, you need to also know how to do it. And so that is why we have lectures in the morning, but then the do component in the afternoon. And so you should realize that that is where you get most out of this exercise. So we have tutorials, as you heard already from Yvette, that are available here. We didn't print out every possible tutorial. We have over, in Urbana, just in Urbana, we have over 1,000 typeset pages of tutorials. And so we couldn't bring it all. So if you have a question, just ask, and you might be able to download it, and maybe we can even print it out here for a few of you. I don't know what the possibilities are, otherwise you just have it on your computer. Help each other, that would be very good. I said already, model your own system and ask us about it. And very important for us is that we evolve the concept of the workshop. So at the moment we try to start a new way of doing them. Just they were very successful, but somehow I'm afraid if you don't evolve something, it dies. And so we want to push it further. And of course, the most important input we can get is input from you guys. And so we are not nice to ourselves in asking you the question, which lecture do you like and which tutorial do you like? We always, when we are getting the results from the questionnaire, we're always nervous, you know, oh my God, did I foul up this time? And what will they tell about me? But being so direct is very important. And we really ask you, you know, be frank with your comments and help us thereby to improve what we are offering you. On the other side, we also like positive feedback, of course, but that's more like in the political plane, that if we are getting good feedback, then we can also say, you know, this is a worthwhile effort. We put a lot of funds in it. In this case, actually, it is mainly funded by Pittsburgh or by Pittsburgh colleagues. But when we're doing it in Urbana, then we are putting funds in it. And I think we need about $30,000 at least for each workshop in funds. And so we have to ask always for the money. And then, of course, we have to tell then NIH back that the workshops are well received and doing well. So we both need from you, you know, like an arm length kind of description. Yeah, this was really helpful, but also in detail, we like the description of where you think we can do better. So even though Yvette thanked already everybody, I like to again thank the teaching assistants they are putting in always a hard week here, and so I say that more now also, that you ask the teaching assistants about your questions. Juan is a postdoc in my group. He's actually the main person behind the HIV project. When Ma is a graduate student in my group, he does some fabulous work on molecular motors, particular hexamerically case. So he's really between the conceptual mathematical side and computing. And Daniel Chandler is a postdoc in my group, also turning to the position of assistant director of our group. And she is an expert in photosynthesis, and she is doing simulations that make us understand the process of photosynthesis better. And so these are the people for me, you know, a little bit their background. And that's it. Now, what I would like to